Okay, we're going to talk about genetics today, which is chapter 9 from your book. These slides are from the lecture that's posted. I just cut some of them out. But the first thing you have to be familiar with are a couple of terms. First of all, we all have two sets of what are called alleles. And alleles are varieties of genes. So the gene is the actual trait, the character that we're looking at. So, for example, Gregor Mendel used pea plants that had flowers. So the trait he looked at was flower color, and the alleles were purple and white. So a gene is the specific character that you're looking at, and the alleles are the varieties of those genes. So in humans, for example, hair color would be a trait, a gene. We have a gene for hair color. And then blonde, brown, black, red, those would all be alleles. So we get two sets of alleles, one from mom, one from dad. Another term, genotype versus phenotype. Genotype are the actual genes that you inherit. So the physical alleles that you inherit. Phenotype is your physical appearance, what it looks like. So for example, again with pea plants, we'll go with Gregor Mendel looked at flower color, and Gregor Mendel was the first one to actually do all of these studies in what we now know as genetics. So he used pea plants and looked at flower color. There are purple flowers and white flowers. So what he discovered was that purple flowers are what we call dominant, white flowers are what we call recessive. So I already said dominant is expressed whenever it's present. Recessive is expressed only if both alleles are recessive. So purple is dominant, white is recessive. So if we're going to look at the genotype of these plants, it would be uppercase P, uppercase P, uppercase P, lowercase p, or two lowercase p's. Those are the three options, right? We have a dominant trait and we have a recessive trait. So you can have both dominant alleles, you could have both recessive alleles, or you could have one of each. So since the dominant is what is expressed when it's present, the two uppercase P's, the two dominant alleles, are going to be purple, and the dominant allele and the recessive allele together are going to produce purple flowers. But the white flowers are two lowercase p's, so both recessive alleles. Both recessive alleles have to be present for that recessive to be expressed. So Gregor Mendel used some techniques. True breeding is when a plant is self-fertilized and all of the offspring are identical to the parent. So if you have a purple parent and all the offspring are purple, for example. If you have two different varieties, they're called hybrids. So if you have a dominant and a recessive trait, and that cross-fertilization is a genetic cross, and that's what we're going to be doing for your genetics assessment. So the parental plants are named the P generation, P parental. The offspring of those parental plants are called the F1 generation. And if you cross the F1 generation, you produce an F2 generation. So these are the traits or characters that Gregor Mendel looked at: flower color, flower position, seed color, seed shape, pod color, and stem length. You can see the dominant traits versus the recessive traits. So let's go with the flower color. Dominant is purple. We know that the recessive has to have two alleles in order to be present and show through. So all of those white flowers, their phenotype is white. Their genotype is going to be lowercase p, two recessive alleles. The purple flowers, that's the dominant phenotype, but the genotype could either be uppercase P, or uppercase P, lowercase p. So for the dominant phenotype to be expressed, you could have both dominant alleles or one dominant, one recessive allele. When you're doing crosses, you usually use the first letter of whatever the dominant allele is. So in this case, purple would be P's. In seed color, for example, yellow is dominant, so we'd use Y's. For seed shape, round is dominant, so we'd use R's. So when you cross two individuals that are different in these single characters, it's a monohybrid cross. Mono means one. So Mendel performed a monohybrid cross between a purple flower plant and a white flower plant. Now we know the genotype of that white flower plant has to be little p, but that purple flower plant could either be big P or big P little p. The F1 generation produced all plants with purple flowers. When you crossed the F1 plants, they produced a generation that were 3/4 purple and 1/4 white. So what does that mean? Well, here we go. True breeding purple means that it is two dominant alleles. So uppercase P, uppercase P. A true breeding white flower, the only option is lowercase p, because that's what it has to be for those white flowers to be shown through, because they're recessive. So the F1 generation produced all purple flowers. Because the purple flower true breeding parent plant is two uppercase P's. So it only has an uppercase P to donate to the offspring. In turn, the white flowers only have a lowercase p to donate to the offspring. So that means that all of the flowers for the F1 generation are going to be big P, little p. Now that's what we call homozygous versus heterozygous. Homozygous, the purple flowers, let's say, homozygous means they have the same allele for both. Both alleles are the same. So it would be big P. Homozygous recessive, of course, has to be little p. And heterozygous means one dominant, one recessive allele. So big P, little p. So if you cross homozygous parents, a homozygous dominant with a homozygous recessive, all of the F1 generation are going to be heterozygous. Because again, purple can only donate the purple allele, white can only donate the white allele. So the F1 generation will have one purple allele and one white allele. But since purple is dominant, they will all be purple. When you cross the two F1 generations, now you're crossing big P, little p times big P, little p. So now they each have an uppercase P to donate and a lowercase p to donate. So the F2 generation, 3/4 of them are going to have purple flowers and 1/4 of them will have white flowers. However, the genotypes of those purple flowers will be different. So the all-purple F1 generation did not produce purple flowers as predicted by the blending hypothesis, which the blending hypothesis basically stated that if you cross two different alleles, you get something in the middle. So Mendel had to explain why the white color wasn't apparent in the F1 generation and why it reappeared in the F2 offspring. So he developed 4 hypotheses, and these are described using what we know now. So alleles are the alternative versions of genes that account for variations in inherited characters. So again, the gene is the actual trait you're looking at, so flower color in this case. The alleles are the versions of that trait, so purple versus white. The second one was for each character, we inherit 2 alleles, one from each parent, and then the alleles can be the same or different. So this is what I just explained. Homozygous genotype has identical alleles, so 2 uppercase P's or 2 lowercase p's. A heterozygous has 2 different alleles, so an uppercase P and a lowercase p. If the alleles of an inherited pair differ, then one is going to determine the organism's appearance, and that's the dominant allele. So as I said, the recessive allele has to be present for both alleles in order for it to be expressed. If the dominant allele is present, that's what's going to show through. So the phenotype is that appearance, the expression of the trait, so purple versus white. And the genotype is the genetic makeup of that trait. So uppercase P, lowercase p. So the same phenotype can be determined by more than one genotype, meaning that 2 uppercase P's produce purple flowers, and an uppercase and a lowercase p also produce purple flowers. And lastly, a sperm or an egg carries one allele for each inherited character because the allele pairs separate from each other during this production of gametes. So gametes are our sperm and our egg. They only have half the number of chromosomes that the zygote has, or the adult has, because when they merge together and fuse together, it restores the whole. So in humans, for example, sperm and egg have 23 chromosomes, so that when they fuse together, they create 46, which is a normal adult. But this statement is called the law of segregation. So again, gametes fuse together and create the pairs again. His hypotheses also explain that 3-to-1 ratio because the F1 hybrids all had that big P little p genotype. So a Punnett square is what we use to show the possible combinations of alleles that can occur when these gametes combine. And that's what you're going to be doing in your genetics assessment. You're going to be creating Punnett squares, and you're going to be answering questions like, what is the expected ratio, and why, and what is the dominant versus recessive. So when we look at the gametes, purple versus white flowers in the parental generation, as I said, the purple is homozygous, so it only has the uppercase P. The white is homozygous recessive, so it only has that lowercase p to donate. So when the alleles segregate, they're going to produce all big P, little p. When those alleles segregate from the F1 generation, you're going to have half be big P and half be little p. So when you put a Punnett square, which is the F2 generation down there, sperm can have a big P or a little p, and the eggs can have a big P or a little p. And then all you have to do is match up for each square. So the first square is 2 uppercase P's. The second one is an uppercase and a lowercase. The third one is again an uppercase and a lowercase. And that fourth one is 2 lowercase. So the phenotypic ratio are 3 purple to 1 white. But the genotypic ratio is 1 homozygous dominant to 2 heterozygotes to 1 homozygous recessive. And that's an important distinction. So your phenotypic ratio, what we see, the appearance is 3-to-1, but the genotypic ratio is 1 to 2-to-1. A dihybrid cross is when you mate 2 different characters. So he looked at round seeds that were yellow and wrinkled that were green. Round and yellow are dominant, wrinkled and green are recessive. So all of the plants of the F1 generation had round yellow seeds. However, they were heterozygous for those because just like the dominant purple and the recessive white could only donate those specific alleles, it's the same thing here. So round seeds can only donate that round. Yellow can only donate that yellow. So you'd have a big R and a big Y. Wrinkled can only donate the wrinkle, and green can only donate the green. So you'd have little r, little y. So all of the plants with round yellow seeds in the F1 generation would be big R, big Y, little y. The F2 generation is going to end up producing seeds in a 9 to 3 to 3-to-1 ratio. 9 are going to be homozygous dominant. 9 are going to be round yellow seeds, so both dominant traits showing through. 3 are going to have wrinkled yellow seeds, so 1 dominant trait, 1 recessive. 3 will have the other dominant and 1 recessive traits, so round green seeds. And 1 out of 16 will have both recessive alleles, so wrinkled green seeds. And how do we do this? Well, we look at the round yellow dominant and the round or wrinkled green recessive. So 2 big R's and 2 big Y's versus 2 little R's and 2 little Y's. So again, the gametes formed are big R, big Y, little r, little y. So that the F1 generation is going to be heterozygous for both. Now this is where it gets a little tricky. When you do your Punnett square, it's a dihybrid cross. So because it is 2 traits, and because the F1 generation is heterozygous for both traits, the sperm and the eggs can produce 4 different gametes. So you can have big R, big Y, little r, big Y, big R, little y, or little r, little y for both egg and sperm. Then when you cross those, if you look, 2 big R's, 2 big Y's, that's going to be round yellow. Big R, little r, big Y, again, round yellow. So the whole first row is basically round yellow. And that's because one dominant trait is present in both characters. So you have a big R and a big Y present all the way across the board. The second row, that first one, again, round yellow, because you have a dominant round and a dominant yellow. That second box in the second row, though, is wrinkled yellow, because you have 2 recessive wrinkled, and then the dominant yellow. And if you go down and look, this is how it kind of goes. So you end up with 9 out of the 16 that have the dominant yellow and the dominant round. You have 3 out of 16 that have the recessive green, but the dominant round. Then you have another 3 out of the 16 that have the dominant yellow, but the recessive wrinkled. And only 1 out of 16 that ends up with both recessive traits. So this is how his hypothesis is supported. So anytime you're asked if you have a heterozygous dihybrid cross, what are the phenotypic ratios? It is 9:3 to 3:1. And that's phenotypic, not genotypic. Genotypic, it's a lot uglier and you'll figure it out on your assessment. But phenotypic, it's 9:3 to 3:1. And for genotypic, you just count how many big R, big Y's there are, how many big R, little r, big Y's there are, and just write it up. So he had new non-parental combinations of traits and had this 9:3 to 3:1 phenotypic ratio. So he suggested that inheritance of one character didn't affect the inheritance of another. So a dihybrid cross is actually equivalent to 2 monohybrid crosses. So this was named the law of independent assortment. So each trait assorts independent of the other traits. So that's how you can get non-parental combinations. So now we're going to look at the independent assortment in Labrador Retrievers. We're going to look at 2 characters, color and vision. So you have black coat is dominant, normal vision is dominant, chocolate coat and retinal myopathy basically is recessive. So to have the chocolate coat and be blind, you have to have both recessive alleles for both traits. So little b, little n. For black coat normal vision, you only have to have 1 dominant of each allele. And then the other 2 kind of flip-flop. So for black coat, you have a dominant allele, but blind, you have 2 recessive. For chocolate coat, you have 2 recessive alleles, but normal vision is 1 dominant allele. So when you cross 2 heterozygotes for these traits, their genotypes are big B, big N, little n. Their phenotype is black coat, normal vision, because they have those dominant alleles. But again, when you cross them, you're going to have 4 gametes for sperm, 4 gametes for eggs. Those gametes would be big B, big N, little b, big N, big B, little n, little b, little n. Sorry, I had to think for a minute. And you can match them up however you want. You can do big B, big N, big B, little n, little b, big N, little b, little n if you want to do it the easy way. You know, match the big B up with each and then match the little b up with each. That's the easy way. But you're going to have 4 alleles for each. So your phenotypic ratio is going to be 9:3 to 3:1. Test crosses are matings between an unknown genotype and a homozygous recessive. So basically, they can show you what that unknown allele is. So you use the homozygous recessive because you know their genotype. Homozygous recessive has to be 2 lowercase letters. There's no other genotype it could be. So let's go back and let's just look at the purple versus white flowers. So we know white flowers are 2 lowercase p's. We know that. But the purple flower, we're not sure if it's homozygous or heterozygous, because as long as it has that one dominant allele, it's going to be purple. So Mendel used test crosses to verify that he had these true breeding varieties of plants. So if we have a chocolate lab, we know their genotype is little b, little b. If we have a black lab, we're not sure what the black lab's genotype is. We just know it's one big B. So that black lab could be big B, or big B, little b. Now, if you do a test cross, if that black lab is big B, all it has to donate is a big B. Now we know all the chocolate lab has to donate is that little b. So if the black lab is in fact a homozygous dominant, all of the offspring will be black. They'll be heterozygotes, but they'll be black. If the black dog is a heterozygote, then he's got 2 alleles to donate, a big B and a little b. The chocolate lab still only has 1 allele to donate the little b. So what we would end up is with half black labs, half chocolate labs. So a test cross can confirm the genotype of an unknown. Because, again, if you end up with all black offspring, it was a homozygous dominant. If you end up with half black offspring, half chocolate, it was a heterozygote. Pedigrees show inheritance patterns if you look through multiple generations. So it's going to either be dominant or recessive inheritance, and it can also be used to figure out genotypes of different family members. So this is what a pedigree looks like. Females are circles, males are squares. The clear circles or squares have the dominant trait. Colored in shaded circles or squares have the recessive trait. So in this case, it's widow's peak is dominant, straight is recessive. So if you look at the first generation, you have Al and Beth that have widow's peak alleles. You have Charles that has a straight hairline, and Debbie that has widow's peak. Now Charles has the recessive hairline, so we know Charles is little h. So if we look at their offspring, Isabel and Juliana. Juliana has the straight recessive alleles. So she's little h. That means that she got 1 little h from her dad, Charles, which we knew. But she also got a little h from Debbie, her mom. So that means that Debbie has to be a heterozygote. She has to be big H, little h in order for Juliana to have that recessive allele. Then when you're looking at Al and Beth, 2 children, Frank and Gary, have the recessive allele. So that means that both Al and Beth have to be heterozygotes. They each had to donate a recessive allele in order to have children with the recessive traits. Okay? And there it is, all filled in for you. Now, not all genotypes can be determined. For example, Evelyn over there. We know that Al and Beth are heterozygotes, but we don't know if one of them gave Evelyn the dominant trait or if they both give Evelyn the dominant trait. Because Evelyn doesn't have children, so we can't see what her offspring look like. So she could still be either or. So just because a trait is dominant doesn't mean that it's more common than a recessive trait or normal. I'll give you an example. 6 fingers is actually dominant. But how many people do you know that have 6 fingers or toes? Not many. Over time, it was basically selected against. So even though it's dominant, doesn't mean it's the most prevalent in a population. Wild type traits are not necessarily specified by dominant alleles. These are just what we see out in nature. So some genetic disorders are controlled by dominant traits, some are controlled by recessive traits. These disorders show inheritance patterns like the traits that Mendel studied. So you guys should be familiar with all of these disorders. Albinism, cystic fibrosis, PKU, sickle cell, and Tay-Sachs disease are all recessive disorders. That means that they have to have both recessive alleles to inherit these disorders, which means that both of their parents have to be carriers. Dominant disorders, though, achondroplasia, Huntington's, and hypercholesteremia, it's dominant disorder. So they only need one allele to have this disorder. So one parent cannot be a carrier at all. And if the other parent has this disorder, they can pass it on. Now it'll depend, because if the parent is a homozygote dominant, then they're definitely passing the disorder on to their children. But if they're a heterozygote, there's a chance that they'll pass the recessive allele on to their children and may have normal children. Human genetic disorders can range from mild, such as albinism, to fatal, such as cystic fibrosis. Cystic fibrosis basically causes a mucus buildup in your lungs and respiratory system, and oftentimes you can't breathe and you need oxygen and you have to have your lungs kind of pumped almost, and the life expectancy is about 35 now, which is good from what it used to be. So most people who have these recessive disorders are born to normal parents who are just carriers. So their parents are phenotypically normal, but they're carriers of this recessive disorder. So here you have two normal parent carriers. They can donate either the dominant or the recessive allele. So you're going to end up with 25% normal children, 50% will be carriers, and 25% will have the disorder. So as I said, CF is lethal. Again, it's about 35 now. The recessive disorder carried by about 1 in 31 Americans. Again, you have an excessive secretion of this mucus from the lungs and other organs that causes this unbelievable buildup to where the lungs almost have to be pumped at times. Dominant, like Huntington's disease, it's a degenerative order of the nervous system. And Huntington's disease usually doesn't manifest itself until about 30 to 40. But within 15 years, unfortunately, you die because your nervous system just gradually degenerates over time and your brain starts to degenerate. Achondroplasia is a form of dwarfism where the head and the torso develop normally, but the arms and legs are really short. And again, these are dominant disorders, so they only have to pass on one allele to pass on the disorder. So we've been looking at what are called complete dominant situations. One dominant allele, one recessive allele. We also have certain phenotypes where it's an incomplete dominance. So they're not totally dominant one over the other. Snapdragons, for example. You have red and you have white. Red can donate the big R, white can donate the little r. But the F1 hybrid generation is actually pink. So it's an intermediate of the two, big R, little r. So you might say that this would support the blending hypothesis. However, when you cross the F1 generation, those white flowers come back in 1/4 of the offspring. So that's how we know the blending hypothesis still isn't supported, because the offspring in the F2 generation are 1/4 red, half pink, and 1/4 white. So that white color makes an appearance. Hypercholesteremia is an example in humans. You can have really high levels of cholesterol in the blood, but heterozygotes only have an intermediate high cholesterol level. So if you're a homozygous dominant, your cholesterol is off the charts, but if you're a heterozygote, your cholesterol is high. So this is just showing you homozygotes have really high, off the charts cholesterol, heterozygotes not as much, and It's a recessive, it's an incomplete dominant disorder. So homozygous dominant are going to have normal cells, heterozygotes mild disease, and homozygous recessive are severe disease. So every individual carries 2 different alleles for a particular gene. Sometimes if you have multiple alleles, though, more than 2 possibilities can exist, like blood typing. Human blood have the ABO groups. So we have A, B, AB, and O. Now, A and B alleles are expressed at the same amount in the heterozygote individual, so that means these alleles are codominant. So incomplete dominance means you have an intermediate kind of blending of the 2 homozygotes. So like in the hypercholesteremia, you have normal, you have severe, and then you have in the middle. So that's incomplete. Codominant means they're both expressed equally. So on the AB blood type, for example, the A and the B are both expressed. So here you can have AB, AB, or O. Whatever genotype you are, you have the opposite antibody in your blood, which is why you can't get blood transfusions from a different blood group. So for the genotypes for A, you could have 2 A's, or an A and an O, basically. For B, 2 B's, or a B and an O. But for AB, you have an A and a B, and they are equally represented in your blood. O, you have 2 O's, basically. So if you're A, you have anti-B, which means you will react if you get B blood. If you have B, you have anti-A, so it'll react if you get A. AB, you don't have any antibodies, because you have both A and B antigens. So if you had antibodies, they would attack your own blood. So you don't have any antibodies. However, if you're O, you don't have the carbohydrates A or B. You don't have either antigen. So that means you have both antibodies. So AB is the universal recipient because it can get any type of blood because it doesn't have any antibodies. And O is a universal donor because it doesn't have any antigens to get a reaction from your body. So if you're type A, you can get A blood or O. If you're type B, you can get B or O. If you're AB, you can get A, B, AB, or O. But if you're O, you can only get O. And the side is just showing you the clumping reactions to tell what blood type it is. Sex-linked inheritance is the last one we're going to talk about. This means that they're located on the sex chromosomes. The X chromosome is the main one, so oftentimes it's considered an X-linked recessive trait. Most of them are recessive if they're X-linked. Fruit flies, for example, can have red eyes or white eyes, and that's on the X chromosome. So a female with red eyes could be X big R, X big R. A male with white eyes is going to be X little r, Y, because remember, female are XX, males are XY. So when you cross the sperm, you could have X little r or Y, but the female only has X big R to donate. So when you do the cross, you're going to have half females that are carriers, but will have red eyes, and half males with red eyes. So no white eyes at all. Now, if you cross a heterozygote red-eyed female with a red-eyed male, again, the sperm are going to have X big R, Y. Female will have X big R or X little r. Now, this means that all of the females will basically have red eyes. Half of the females will be homozygous, half will be heterozygous. But interestingly, half of the males will have red eyes, and the other half of the males will have white eyes because of that heterozygote female. And then again, heterozygote female with a white-eyed male, you're going to end up with half of your females having red eyes but being heterozygous, half of your females having white eyes, half of your males having red eyes, and half of your males having white eyes. Now, if you notice, it's on the X chromosome. Males only have one X chromosome. So if they get this recessive trait, they have that, whatever it is. Females, however, have 2 X chromosomes. So females have kind of a buffer. So if females have 1 dysfunctional X chromosome, but 1 functional X chromosome, they will be normal, phenotypically speaking, because they have that good X to kind of make up for that bad X. So again, most sex-linked disorders are going to be due to recessive. They're going to be seen mostly in males. A male receiving that single X-linked disorder from his mother is going to have that disorder because the father donates the Y. A female must receive that allele from both parents to be affected. So a female, because she has 2 good X's or 2 X chromosomes, has to have both bad X's in order to be affected. Hemophilia, red-green color blindness, and Duchenne muscular dystrophy are examples of recessive X-linked disorders. Hemophiliacs do not have all the clotting proteins that are necessary to clot their blood, so they will bleed excessively. Red-green color blindness do not have the proper cones in the eyes, so they'll have trouble telling differences between colors. And Duchenne muscular dystrophy, you have a progressive weakening of your muscles, loss of coordination. So again, recessive, males are going to be impacted more than females because males only have 1 good X chromosome, females have 2. So as long as they have that 1 good X, they're fine. So this is the first thing on your assessment. So what you need to pay attention to is the gametes that are produced. 4 cells are going to be produced. And remember, gametes only have 1 chromosome in them. So for the first one, for example, big A, you're going to have 4 gametes that are produced, but each one only has 1 gene or allele. So it'll be big A, big A, big A, and big A. Okay? And then you can figure out the other 2, hopefully. This is the next one. During fertilization, 2 gametes fuse. So it shows you the gametes. 2 little a's are going to create what diploid genotype? Little a. And then you can put the other ones together. And for the last example, I'm showing you a Punnett Square. So basically, if you have a white flowered plant, you know that's recessive. So that's going to be 2 lowercase p's. So the gametes of that white flowered plant are going to be little p. Now the purple flowered plant can either be big P, or big P, little p. And you have to kind of draw the Punnett square to figure out based on the offspring that you have. So if it's big P, that can only donate the big P. So all of your offspring are going to be big P, little p with purple flowers. However, if it's a heterozygote, you're going to have a big P and a little p. So that changes it, because if you have your 2 little p's for your white flowers, and then your big P and your little p for your purple flowers, you're going to have half that are big P, little p, but purple still, and half that are 2 little p's and white. So hopefully this helped you out on your assignment and not only understanding genetics. If you still have questions, of course, send me a message. Otherwise, I will talk to you later.